Obrigado, Dr. Esper, pelo convite. Eu amo ele. Ele é um homem legal, médico, cientista, ótimo. Amo ele muito. Desculpe, Esper, nosso segredo está fora. Me perdoa, sou escocês. Sotaque escocês é muito ruim. Actualmente, a gente pensa, eu não falo inglês. Por causa disto, eu preciso falar em inglês e não falo português. Desculpa. So, vamos. So I'm going to speak about obstacles to curing HIV. So up until recently, the word cure has been verboten, forbidden in HIV research circles. But for some reason, scientists have embraced the notion that we need to work harder to try and identify ways to eradicate HIV from individuals who are infected. So I'm going to talk about some of what I think are some of the major obstacles to curing HIV and suggest some strategies that might help us go forward. And the problem is familiar to many of you. So this cartoon illustrates what happens when individuals interrupt therapy. So, under antiretroviral therapy, viremia is undetectable. Individuals can remain on antiretroviral therapy for many years. The virus is undetectable. But in every single individual who has interrupted treatment, we see this rapid return in plasma viremia. So the virus has established a way to maintain persistence in the host despite this overwhelming antiretroviral therapy. And the big question facing many researchers in the cure field is how HIV is able to persist in the face of antiretroviral therapy. And this is a very contentious issue. Actualmente, cientistas comportamento dos cientistas parece telenovela. Muito drama, lágrimas, and really it's because there are quite strong opinions in what are the obstacles to curing HIV, and as a consequence, what are the strategies to remove those obstacles. So here are three of the, I suppose, the major hypotheses for how HIV is able to persist in the face of therapy. At the top is a model that is based on viral latency. Latency is a term used to describe a cell that is not making the virus, but has the capacity to do so if conditions present themselves. And then under that is a model known as homeostatic proliferation, pioneered by Rafik Sekeli and colleagues, and he's going to be here presenting his work later in the course. And what Rafik is maintaining is that the cells that harbor the virus in a dormant form proliferate, and when the cell divides, the cellular DNA and the proviral DNA is duplicated at mitosis. And some of those duplicated proviruses will simply deplete through attrition, but the overall effect is to maintain the population of latent viruses. And a less popular model, now these aren't mutually exclusive models. I'm not saying that one takes precedence over the other. They may all be acting in concert. But a less popular model is that the reservoirs of the virus are perpetuated by replenishment. that even in the face of overwhelming antiretroviral therapy, there may still be some residual replication that replenishes and tops up those long-lived reservoirs. So I'm going to focus on the issue of ongoing viral replication. Now, although on one side we have latency and the other side we have ongoing viral replication, there are important predictions that stem from each of those mechanisms. So, on the side of latency, there are certain predictions based on the belief that antiretroviral therapy completely stops all ongoing viral replication, so that individuals on suppressive antiretroviral therapy have no residual replication. The virus is stopped dead in its tracks. And under this model, the stability of those reservoirs is dictated by the intrinsic stability of the infected cell, the resting memory CD4 cell. That, of course, provides the immune system with immunologic memory, the ability to respond to antigens that one encountered in childhood and respond to them in adulthood. Now, an important prediction of latency and the absence of ongoing replication is that it doesn't matter what you do to the antiretroviral regimens. If you intensify the regimens, it shouldn't make any difference, because when the virus is latent, it is not accessible to the antiretrovirals that we use in the cocktails for the management of HIV infection. So on the side of residual viral replication, the underlying tenet, is that antiretroviral therapy is not completely suppressive, and that there is some residual replication that's escaping the antiviral forces being levied against it. And under this hypothesis, there is some topping up, there is some replenishment of the reservoirs that preserves their continuity. But an important prediction of this hypothesis is that if one goes in with additional classes of antiretroviral agents, one may be able to perturb the reservoirs. Because if there is residual replication, that residual replication may be modifiable, may be impactable by treatment intensification. So at CROI 2013, that was held recently in Atlanta, Hiroyo Hatano and colleagues presented data from an intensification study called the SCOPE trial, involving intensification with an integrase inhibitor called raltegravir. Now, the last time I spoke here, I discussed results obtained through a collaboration I was involved in with a group in Barcelona, Javi Picado, Bonaventura Cloté and colleagues, which is actually the exact same design, using raltegravir intensification to ask if you add an additional inhibitor to existing baseline antiretroviral therapy, do you change anything? Do you change any viral parameters, or do you change any host parameters, to give an indication that maybe there is some degree of residual replication? So both studies had pretty much identical design. The Hatano study used subjects on suppressive antiretroviral therapy who had been aviremic for at least 1 year. The majority of patients on average were aviremic for over 3 years. So these are very well-controlled patients. Raltegravir intensification was followed by sampling at 1, 2, 12, and 20 weeks, 24 weeks post-intensification. And at those intervals, Hatano and colleagues analyzed viral cDNA, and specifically total viral cDNA and 2LTR circle DNA, or episomal DNA, by a new PCR technique called droplet digital PCR. Droplet digital PCR in many ways supersedes existing real-time PCR because it doesn't use standards to obtain copy number of a sample. And as you don't have to rely on samples, then it's easier to compare data from different days and actually between different groups. And I think droplet digital PCR is going to really move the reservoir field forward a little bit more smoothly, helping laboratories to compare results. So why would one focus on measurement of episomes? This is a generic throughput of a retrovirus. So the viral particle contains 2 copies of genomic viral RNA that are both used in the reverse transcription reaction to make a cDNA. And I wonder if I can, there we are. The linear cDNA. The linear cDNA migrates to the nucleus. Now, what's supposed to happen is that the linear genome integrates into cellular DNA to form the provirus. But some of those linear molecules are captured by the DNA repair machinery of the cell and converted to circles here. Now, if one applies raltegravir to this situation, raltegravir is an integrase inhibitor. It blocks the integration, it blocks this step, and as a consequence, those linear molecules are exposed to the DNA repair machinery. And in the presence of raltegravir, the frequency of the episomes increases. But this can only happen when a cell is being infected. So what happens in patients? So on the left-hand side are the results from the Boson study published in Nature Medicine. And these are individual profiles of the change of the dynamics in the frequency of 2 LTRs following raltegravir intensification. And basically what you're seeing is a sharp increase in the frequency of 2 LTRs following raltegravir intensification and a subsequent decline to baseline. And here are the results of the Hitano study on the right. Essentially identical results obtained with a different cohort of patients on different baseline antiretroviral regimens. The difference actually is that in the Boson study, about 30% of the patients who underwent raltegravir intensification exhibited a dynamic increase in episomes. In the Hitano study, it was 50%. So a higher percentage of patients responded. But all other aspects of these studies are superimposable. The increase was more likely to be observed in individuals who were on protease-containing regimens. So what is the explanation for this impact of raltegravir? The only explanation, the only scientific explanation, is that these individuals, despite the fact that they were on highly suppressive regimens and that they were suppressed in terms of their viremia, still had some degree of residual infection occurring somewhere in their bodies. Because for episomes to increase upon raltegravir, one first needs infectious viral particles to deliver the substrate for the episome, which is the linear cDNA. And the linear cDNA has to be generated by reverse transcription. So you need both infectious virus and you need reverse transcription. Things that technically should not be happening if one is on highly active antiretroviral therapy containing protease inhibitors and reverse transcriptase inhibitors. So one possible explanation is that perhaps some of the antiretrovirals aren't reaching all the locations in the body where HIV-1 is residing. And we know a lot about the pharmacokinetics, the behavior of antiretrovirals, mainly from the study of peripheral blood. But there is far less known on how antiretrovirals permeate the tissues, in particular lymphoid tissue, which is the predominant site of viral replication. So in collaboration with Ashley Haas, Tim Shacker, Courtney Fletcher, and others, we intended to look within lymphoid tissue for whether there was some suboptimal virologic response to antiretroviral therapy that could explain why raltegravir is able to impact episome dynamics in patients who are otherwise completely suppressed. And this involved baseline and sequential sampling over 6 months in gut and lymph node tissue from patients initiating suppressive antiretroviral therapy, followed by examination of viral cDNA intermediates, proviruses, and 2LTR circles, as well as cell-associated RNA, and more importantly, intracellular levels of antiretrovirals. And for example, here we have an individual who initiated antiretroviral therapy, and as one would expect, there was a precipitous decline in plasma viral RNA, so the virologic response appeared to be as one would expect for someone who was viremic and going on therapy. However, if one looked within lymphoid tissue at the frequency of these extrachromosomal forms, there appeared to be a suboptimal virologic response. Rather than episomes declining throughout the 6 months of initiating antiretroviral therapy, there was an increase in the episomes relative to baseline and month 1 time points. And then Courtney Fletcher examined intracellular drug levels in various compartments, including lymph node, ileum, and rectum. And if you focus your attention on lymph node, for example, one can see that there are very low concentrations of some of these antiretroviral agents compared to peripheral blood, in particular atazanavir, darunavir, and efavirenz. So despite suppressive antiretroviral therapy, there is some de novo infection occurring in lymphoid tissue, and especially in lymph node. One possibility is that there is an inability to sequester antiretrovirals in tissue lymphocytes that may create conditions for episodic rounds of viral replication. And obviously those findings have implications for strategies aimed at reducing the persistence of the viral reservoirs. But how do we reconcile those observations with everything we've begun to learn about HIV and antiretroviral therapy? We know that antiretroviral therapy works. If the individuals are adherent, there is very little, if any development of resistance. Yet here we're seeing that even in the face of antiretroviral therapy, there is some residual replication. So the two observations don't appear to reconcile very well. And if one looks at the literature, there's no impact of intensification on residual viremia. That was observed in both the Hitano and Buzon studies, but it's been observed in many other intensification studies using raltegravir or other agents to intensify treatment. No obvious impact on viral DNA levels. I don't find that particularly surprising because we know that most of the viral DNA in infected individuals is non-functional, what we commonly refer to as graveyard sequences. And those graveyard sequences tend to occlude what might be happening to the population of biologically active proviruses. No obvious impact on cell-associated viral RNA, and a lack of obvious sequence evolution. But of course, we have to treat phylogenetic analysis of viral cDNA obtained from these reservoirs with caution, because one could have some low degree of viral replication, but simply not see any sequence evolution because of sampling. So the fact that there isn't any obvious sequence evolution does not rule out the possibility that there is still some degree of replication and reverse transcription-generated errors. But another way to explain or reconcile these observations is to invoke a model of a chronic virus source. So when we talk about ongoing viral replication, and I think this is the term that creates more angst, that creates more disagreement, because virologists, when they say ongoing viral replication, this is the sort of thing they're thinking about. We have a cell that becomes infected. That cell manufactures viral particles that go on to infect another cell. That cell manufactures viral particles that then go on to infect another cell, so on and so forth. This is what we commonly refer to in virologic terms as ongoing viral replication. And actually, none of the data that I've presented, none of the data from the Hitano study, from the Buzone study, actually convincingly demonstrate that there is ongoing viral replication. Alternatively, one could invoke this model, where there is a chronic virus source. Cells are manufacturing viral particles, but those viral particles are limited in their ability to establish infection in other cells. So there are single rounds of infection where a cell that becomes infected here does not pass the virus on to other cells. So there's not multiple rounds of infection. So what's happening in antiretroviral therapy in lymphoid tissue? One possibility is that antiretrovirals are, for reasons that are unclear, not efficiently sequestering within those lymphoid tissue sites that are the predominant sites for viral replication. So existing antiretrovirals are not accessing these sites of viral replication, and we have these episodic transfers of virus to neighboring cells, but there aren't any multiple rounds. And then we put raltegravir into the picture. Raltegravir has better pharmacokinetic characteristics that allow it to more efficiently access those sites in lymphoid tissue where the virus is replicating, and the raltegravir prevents these single rounds of HIV infection, and that's why we see this impact of raltegravir on these episomal cDNA forms. And for those of you who like beer, there's an old Heineken commercial. And basically what Heineken was claiming was that this beer could reach parts of the body that other beers could not refresh. So here we have Spock, who's looking very gloomy. His ears are drooping, but he has a Heineken, and his ears perk up. And obviously there are many renditions of this cartoon that you can imagine. I won't divulge all the details. But what I'm suggesting is that raltegravir may be an example of Heineken, that it's reaching parts of the body, it's reaching reservoirs that some of the other beers aren't reaching as effectively. A simplistic hypothesis, but one worth exploring. Now, this residual infection, there are people who are more and more embracing the idea that there may be residual infection because of incompletely suppressive therapy. But the knee-jerk response is, it's not relevant because we know that heart works. Well, actually, heart works to a certain level. But it's clear that individuals on suppressive antiretroviral therapy still exhibit comorbidities as a result of the presence of HIV. So, for example, Steve Deek's group has, in a number of studies, demonstrated that CD8 T-cell activation persists in patients on long-term suppressive antiretroviral therapy. And that CD8 suppression can undermine treatment-mediated CD4 gains. And the SMART study has shown that certain comorbidities or markers of comorbidities such as IL-6 and D-dimer are independently associated with an increased risk of cardiovascular disease. So individuals on suppressive antiretroviral therapy still have elevated levels of immune activation, and they still have comorbidities, including cardiovascular comorbidities. And in the Buzone study, one of the more curious accompaniments that paralleled the change in episome dynamics was a normalization of the frequency of activated CD8 cells. So individuals who went on raltegravir and who showed a change in episome dynamics also reduced the frequency of activated CD8 cells to baseline over a period of 24 weeks. And recently we published that if one withdraws raltegravir at 48 weeks, but maintains baseline antiretroviral therapy, one starts to see an increase in the frequency of activated CD8 cells. Raltegravir does not have a direct impact on immune activation status. Raltegravir is an antiviral agent. So the only way that raltegravir could be impacting CD8, the frequency of activated CD8 cells, is through its impact on residual viral replication. And then we have the Hitano study. And what they found is that individuals who intensified with raltegravir showed a statistically significant reduction in D-dimer levels, a surrogate of cardiovascular comorbidities. So beyond the possible clinical implications of residual infection, what are the implications for maintenance of the reservoirs? What's the implication for this residual? What could be the impact of residual infection on reservoir maintenance? Well, we're currently struggling with the sobering reality that latency occurs in CD4 cells that are very long-lived. And if one calculates the decay of these, the decay of the frequency of these latently infected CD4 cells, it's been predicted that time to eradication would be in the order of, it's on here, excuse me, in the order of several decades. In other words, it would take lifelong suppressive antiretroviral therapy to allow these latent reservoirs to decay to zero. And that's a very sobering thought that we can't be complacent about, we can't live with. However, if there is some degree of replenishment, then the actual stability of the reservoirs might be much shorter than current estimates based on the reduction in latently infected cells. So if we can stop replenishment, what we might find is a more rapid decay in reservoir size over time. So what fuels viral recrudescence when antiretroviral suppression is interrupted? One can use intensification trials to try and get an idea of whether more intense regimens perturb something, either a viral parameter or an immune host parameter. But another way to do this is to de-intensify and then look at how the virus recrudesces and the tissues that it may recrudesce from. And I just wanted to mention another presentation at this year's Conference on Retroviruses and Opportunistic Infections, presented by Megan Rothenberger. And what this involved was an in situ hybridization analysis of lymphoid tissue in individuals who were on suppressive therapy and who subsequently interrupted their antiretroviral regimen. And basically what that preliminary analysis revealed is that the recrudescence of the virus occurred in lymphatic tissue at multiple anatomic sites simultaneously following treatment interruption. Now, one has to remember that this in situ analysis is a microscopic snapshot of a very large organ. The large and small bowels have an area of 60 and 2,000 square feet. So it begs the question, if this recrudescence is being fueled by an episodic activation of a latently infected cell, how can that reactivation be occurring in these latently infected cells at multiple anatomic sites and simultaneously? I want to finish by just touching on some of the efforts aimed at cure research. I don't want to steal Mattia's thunder because Mattia is going to be giving a much more expanded view of efforts to cure viral latency in HIV-1 infected individuals. So some of the strategies being used are summarized on this slide. But the one I want to talk about is this one here, the use of virus purging agents. So-called shock and kill strategy. You shock the cell that harbors a latent virus to get the virus to wake up, and then you attack that reactivated virus with with antiretrovirals, or you hope that the reactivated virus is going to kill the host cell by cytopathic effect and or by immune surveillance, such as clearance by HIV-specific CD8 cells. And this slide summarizes some of the ways in which scientists are trying to shock the latent virus, the sleeping virus. The reality is that the best way to activate HIVs through the NF-kappa B pathway. The NF-kappa B pathway, unfortunately, there are agents, Vorbol esters, for example, that activate HIV through the NF-kappa B pathway, but we can't use those sorts of agents to shock the virus because of the global impact of such agents on cellular gene expression. You don't want to wake up all cellular genes that are regulated through the NF-kappa B pathway. So a more subtle approach, a more surgical approach, has been to use agents that modify chromatin, shown on the bottom here. Because the virus is lying in a latent state because it's in a hostile chromatin environment. So relieving that chromatin architecture exposes the virus to the sorts of transcription factors that regulate viral gene expression. So David Margolis's group at UNC last year published the results of a clinical trial using vorinostat, or SAHA, an HDAC modifier, to shock HIV out of latency. And the question was simple, whether the questions being asked were simple: is SAHA well tolerated, and does it produce an in vivo effect? And the answers to both questions were yes. The SAHA was well tolerated. Multiple doses were accompanied by very few adverse events. But the level of shock on HIV expression was, in virologic terms, very modest. A 4 to 7-fold increase in cell-associated viral RNA. And as a virologist, I find that quite disconcerting, because we don't know if that 4 to 7-fold increase in viral RNA was actually accompanied by an increase in expression of viral proteins, or whether there was any untoward effect of viral reactivation on the viability of the cell that harbored the latent virus. And if we are trying to shock the virus into waking up and killing the host cell, we need to have those things happen. We need to have viral protein expression for cytopathic effect, but also for recognition by the immune surveillance mechanisms of the host. So it's unclear whether the level of RNA induction equates to expression of viral proteins, and it's unclear whether reactivation results in elimination of the infected cell. And another issue facing the purge, the shock and kill approaches is that they are very T-cell centric. They are geared towards purging a reservoir of latent CD4 cells. And most of the attention on viral eradication strategies have focused on this latent CD4 pool. However, the macrophage, an important target cell for HIV infection, has escaped attention with these approaches. And if macrophage are a reservoir, and that's a big if, there's no data as yet, hopefully there will be soon, that macrophage are in fact holding onto the virus in patients on suppressive antiretroviral therapy, then these shock and kill strategies may or may not have a similar effect on latent infected macrophage. So I'm going to show some data. Here is a macrophage latency model that we've developed in my lab. Macrophage actually very effectively recapitulate HIV latency. These are experiments done with primary macrophage. So in the non-stimulated macrophage here, the control, there is very little, if any, viral gene expression as determined by the levels of viral P24 in culture supernatants. If we stimulate through the NF-kappa B pathway, on the left here, the positive control, PHA, then we see very precipitous increase in the level of P24 production from these latently infected macrophage. And similarly with prostratin, another agent that activates NF-kappa B pathway, we see nice induction of HIV from latency. However, if we look at agents that have been shown to reactivate T-cell latency, vorinostat, there is very little of an effect of vorinostat on latent infection of macrophage, valproic acid, or disulfiram. So agents that have been shown to shock HIV in T cells appear to have less of a capacity to do so in latently infected macrophage. So I'm going to finish. I hope I've given some food for thought as to the various obstacles that we face and the reasons for the debate amongst cure researchers. I just want to highlight what I think are some of the really central questions that we have to attack in the next couple of years as we embark on this venture to try and eradicate HIV. Firstly, what is the contribution of residual infection to maintenance of the viral reservoirs that persist in the face of antiretroviral therapy? And I think to do this, what we need to do is in addition to the looking at the impact of intensification on viral cDNAs, we also have to look at its impact on the size of the reservoirs over time to see if reducing this residual infection actually impacts the stability of these long-lived reservoirs. Secondly, what's the origin of viral recrudescence following treatment interruption? Thirdly, is chronicity, the stability of the reservoir, restricted to CD4 cells? Are there other cells, such as myeloid cells, myeloid lineage cells, playing a role in viral persistence? And will their elimination require different strategies? A very important question, do agents that shock HIV in vivo, such as SAHA, actually have an impact on the viability, the persistence of the host cell? And finally, a practical standpoint, what assays can we actually use to gauge the impact of these various clinical approaches on viral reservoir size? The field has been really struggling with this issue because we really are looking for a needle in a haystack, and we need to be using very sensitive assays that measure the virus, but more importantly, that measure the biologically relevant virus, the virus that contributes to viral persistence. So I'll finish by acknowledging the funders, including the Delaney AIDS Research Enterprise. This is a collaboration of scientists with complementary skills who are focused on the cure issue. Funding from the National Institutes of Health, the institutes, the NIAID and NIMH, and support from Merck. And by way of self-reporting, I have been a paid consultant by Merck Pharmaceuticals. And then some of my co-workers at the University of Miami, at the Curso Institute in Barcelona, Ventura Clotet and Javi Picado, at the University of Minnesota, Tim Shacker and Ashley Haas, who's here in the audience today. He stole himself away from the fishing wastelands of the Arctic to be here. At UCSF, Steve Deeks, Mike McCune, and the Dallaire Collaboratory Scientists, and Courtney Fletcher at the University of Nebraska, who heroically measured intracellular drug levels in lymphoid tissue from individuals on suppressive antiretroviral therapy, and contributors to the talk, providing some of the illustrations, Taewok Chun, Nick Chaumont, Megan Rothenberger, and Hiroyo Hitano. And for those of you who are actually interested in reservoir research, I just want to point out that every year, every December, we will be holding a meeting dedicated to reservoir research. It will be held in Miami. And I'd love to see you there. It's a great time of the year to be in Miami. Okay, you run the risk that you may see some of these senior scientists in their swimsuits, in their Speedos. I apologize for that up front, but hopefully you'll be able to come to the meeting, which will, I think, document our progress in understanding these obstacles to HIV cure, and more importantly, coming up with strategies to circumvent those obstacles. So thank you for your attention.
Thank you, Mario. My question is about the varinostat. Because it is not LTR specific. Yeah. Since it is not LTR specific, and if it is going to be to form the heterochromatin, what's going to be happen with the bystander heterochromatin?
So I think the question is the HDAC modelers, are they going to be specific for sites of chromatin that harbor the virus? And what's going to be the impact on other genes that are in condensed chromatin?
Formation.
Well, yes. I mean, the ideal situation is to use reactivation strategies that only activate latent HIV and that leave cellular genes relatively untouched. But when you modify chromatin, you don't have that specificity. It's still a better approach to using NF-kappa B modifiers where you're simply, you're activating many, many genes that have NF-kappa B regulatory elements. So it's not specific for HIV. The chromatin modifiers do not specifically wake up HIV expression. There is collateral effects on other genes that are in condensed chromatin. Does that answer your question?
Yes, sure. And my second question is, if the purging is successful, how much we are sure that the virus that is coming out, we are going to cope up with heart?
I don't think I followed that. Could you repeat it?
If the purging is successful and the virus is coming out, how much we are sure that heart that is available, we are going to cope up with that virus that is coming out with the heart?
The question, I think, is if you purge the virus and it's successful, how are you going to cope with the virus that you've reactivated? Well, heart is going to, obviously you're going to try and purge the virus in the setting of suppressive therapy. Any virus that you drive out of latency, you're going to prevent it from infecting other cells because you have suppressive antiretroviral therapy present at that time to prevent any new cells from becoming infected.
Thank you.
Mara, mainly a comment. I think many of the puzzles in the field, including the current ones, is this long-standing assumption that what you're measuring and looking at in the bloodstream is a dynamic equilibrium and an accurate reflection of what's actually going on in the reservoir sites of production and persistence. That can't possibly be true, as we're increasingly learning. The implications for your last slide, or one of your last slides, is if you're going to look at what these strategies are doing in terms of elimination of the reservoir, you're almost certainly going to have to look at lymphoid tissues as part and parcel of that, however we get at that.
Yeah, actually it raises an important point because, as a field, we use measurements that are based out of convenience. We use peripheral blood as our source, as our clinical sample. But unfortunately, the peripheral blood contributes in a very minor way to the overall burden of HIV reservoir in an individual. And so if we're trying to get a snapshot of what's happening to the HIV reservoir, of the impact of antiretroviral therapy, immunologic modifications, we really need to be looking in the tissues, the predominant sites of viral replication. It's a challenge, but no one said that gaining insight into how HIV persists and, more importantly, how to eradicate HIV was going to be easy.
So if raltegravir intensification increases 2 LTR circles, presumably blocking viral integration events that would have otherwise occurred, could that be an eradication strategy, simply using long-term combinations of drugs that are known to penetrate well into tissues, eschewing, for example, the protease inhibitors and moving to other drugs that are known to get in? Could we go back to sort of a simple idea of just using truly highly active therapy for a long period of time?
I don't think I'm going to stick my neck out that far. But at the very least, I think we need to
Stick something out, yeah.
Well, I don't think we're going to eradicate HIV by limiting residual infection. But to cure HIV, we're going to have to limit residual infection. And I think the next step, the original raltegravir intensification study was controversial. I mean, very few people actually believed it because most of us think that HAART just shuts down all viral replication. The second study, I think, has removed the doubt. And hopefully that will spur investigators into now using these intensification studies to try and basically look at the impact on reservoir size. But there's got to be a commitment. I mean, there's got to be a commitment from the funding agencies as well, because if you look at the impact of intensification on, for example, immune activation, it's not a rapid process. Most people were looking 6 weeks and 12 weeks later for changes in immune activation status. It takes many months to see these changes in immune activation status on raltegravir intensification. So you have to do the study right. And in a long-term, and under a long-term study, with now the assays that we have, such as droplet digital PCR and more effective ways of measuring viral latency, we should be able to see if stopping replenishment, or at least stopping that residual infection, leads to a more rapid decline in the size of the reservoir. And you know, personally, my personal bias is that we will see a more rapid, a more precipitous decline in reservoir size if we can limit residual infection. That's just my bias. Tom.
Mario, I was really struck by that image you showed of the in situ hybridization. And I'm trying to remember the details, but that was of someone on therapy, right? So
Someone who on therapy who interrupted therapy. So you're interrupting therapy and then asking, where does a recrudescence originate from?
right. So if you sort of you told us this is like 2,000 square, whatever the large number there is. How many different samples did you have there, and how often did you see that level?
I think Ashley can
tissue,
answer that better than I
if you
can.
extrapolate that out, how do you relate that to sort of viral load? Is it directly correlated? Because you make very good criticisms of looking at the blood, but usually you're looking at the blood, and so we have to sort of strike a balance. And I think a key thing would be to understand the relationship between what's going on in tissue and what's going on in blood, and maybe we could use that as a guide to better interpret this data.
Tom, what was amazing for the points that you raised is you would expect, and we had anticipated, that we would come up with nothing trying to go through this huge 2,000 square foot house with, I think we looked at between 100 and 150 sections to find, there were several examples in this particular patient. So it is doable. And what was striking was not only the multifocal nature, but the ease at which we consistently found in those patients, in the lymph nodes and the gut samples, these multiple foci. You can explain it with some difficulty with multiple foci of latency and reactivation, but you can explain it a little more easily, I think, if there's some level of, very low level of ongoing replication that you now relieve and that the virus begins to take off, just in terms of the sampling issues that you're raising.
Well, do you know what the serum level of virus was at that point?
Serum level was, on average, under 1,000 copies. So the one model would be that you have a latency, and a latent infection gets reactivated, you have secondary, tertiary replication, then you seed all of the distant sites and get that. That still could be true, but it's also consistent with a little bit of virus production coming in and being summed from those different sources.
And I'm less in favor of a latent infected cell becoming activated stochastically and then fueling all of these foci that then allow recrudescence in a focal nature, because then you would have to have two models for recrudescence, one in which a latently infected cell spread the virus to all of these sites, and then those sites allowed focal recrudescence of the virus. So I think it's easier to explain if you have chronic virus sources that are productively infected, but there's no spread because the virus is being held in check by antiretroviral therapy. So they're like smoldering embers, if you want to use an analogy. In a campfire, smoldering embers, there's plenty of trees around to ignite the forest fire, but the antiretroviral therapy stops that. But again, more work is needed, but the observations are intriguing.
Well, thank you, Mario, again.